Okay, so this week I'm doing my video by myself, but I have here the work for the lesson 7 group problems. So I'll just go ahead and go through them. For number 1, we were given the genomes of 4 different viruses, asked to identify the double-stranded versus single-stranded, whether they were DNA versus RNA, and whether they were circular or linear. So for genome 1, I said that it was circular. This was because it was resistant to exonucleases, meaning that in exonucleases, they require a free 5' or 3' end, which the circular do not have. So I deduced that was circular. And then I said it was double-stranded because the A to U ratio and C to G ratios were about equal to 1. And then I said that it was RNA because it used uracil and not thymine. And for genome 2, it was sensitive to exonucleases and endonucleases, so I said it was linear because it had a free 3' or 5' end. I said it was single-stranded because the A to U ratios and G to C ratio were not equal to 1. And then I said it was RNA because it used uracil and not thymine. Then for 3, linear because it was sensitive to both exo and endonucleases. Double-stranded because the A to T and C to G ratios were about equal to 1. And DNA because it used thymine, not uracil. And 4, circular because it was resistant to exonucleases, meaning it did not have a free 5' or 3' end. Single-stranded because the A to T and C to G ratios were not equal to 1. And then DNA because it used thymine, not uracil. Then for 2, A, I got topoisomerase 2 and nucleosome. For B, 2 and 0.34. C, topoisomerase 1. D, Ori C. E, sulfur 35 and phosphorus 32. And then F, ribose and deoxyribose. So for number 3, it was talking about it says suppose a translocation occurs between chromosome 14 and chromosome 21 in an individual, and it goes ahead and gives us all of the base pair lengths and everything. So the 2 together, so I got the total base pair length from the 2 chromosomes together. So chromosome 14 was 109 million and base pairs, and chromosome 21 was 48 million, so I got 157 million. And then A had asked how long the DNA portion of this translocation chromosome was in meters. So I found its length in nanometers by dividing, I found the number of turns by dividing by 10, got 15,700,000, divided that or multiplied that by 3.4 nanometers to find the length in nanometers. And then I did this conversion to get it to meters, since there's 1 times 10 to the 9th nanometers in a meter. And I got 0.0534 meters. And then B, how many nucleosomes are found within this translocation chromosome? Assume that the average length of the linker DNA is 50 base pairs. So to do this, I found that there were 147 base pairs within a nucleosome, and then 50 base pairs for the linker DNA between each one. And so I added these together to get 197. I divided 157 million by 197 and got 801,020 nucleosomes. And then for C, how many H2A molecules would you expect to find within this translocation chromosome? There are 2 H2A molecules per nucleosome. So I multiplied 801,020 nucleosomes by 2 and got 602,040 H2A molecules. And then for D, how many radial loop domains would you expect to find within this translocation chromosome? And so because radial loop domains are between 25,000 and 200,000 base pairs, I divided 157 million by each of those numbers and got the range of radial loop domains. So 785 to 6,280 radial loop domains. And then for 4, the bacterium Mycobacterium tuberculosis has a single circular chromosome that is 4,410,000 base pairs in length. For A, if the chromosome was stretched out linearly, how long would it be in micrometers? So I divided 4,410,000 by 10 to get the number of turns. And then I multiplied that by 3.4 nanometers, since each turn is 3.4 nanometers, and got the length in nanometers. And then I used this conversion, 1,000 nanometers to every micrometer, to get approximately 1,500 micrometers for A. And then B, what is the diameter of the bacterial chromosome in micrometers? And so I used the circumference equals pi d and put in the circumference as the length in micrometers of the bacteria chromosome, and divided 1,500 by pi to get the diameter, which was 477.5 micrometers, or 478. And C, suppose the bacterial genome is organized into loop domains, with each loop domain containing 50,000 base pairs. If one of these loops was stretched out linearly, how long would it be in micrometers? So I did 50,000 divided by 10, which would be the number of turns, which would be 5,000. And then I multiplied 5,000 by 3.4 nanometers, because 3.4 is the length of one turn, and then got 17,000 nanometers and used this conversion for 1,000 nanometers per 1 micrometer to get 17 micrometers. And then for D, it asks how many histones are found attached to the bacterial chromosome, and the answer was 0, because because it's a bacteria and histones are only present in eukaryotic cells. And then for 5, we were given these 6 different structures, and I went ahead and showed my process. So what I did was I went through and named each of the structures, so I could base it more off of the name than the structure, and so I could double check with the names more likely. And so first, it asks us to identify which of the molecules are nucleosides. So which one of these contain, I wrote it here, which one of these contain just the pentose sugar and nitrogenous base without any phosphate groups? And so as we can see, that would be A right here, E, and F, because B, C, and D all contain phosphate groups. So A, E, and F, and then nucleotides. So these will be the pentose sugars with the nitrogenous base and the phosphate groups. So that'll be the other 3, the B, C, and D. And then ribose. So to identify ribose, I have it written here, but that's going to be the pentose sugar with 2 hydroxyl groups, or a hydroxyl group coming off of the 2' carbon. So that's going to be here C, E, and F. We can see there they have the hydroxyl groups, and then these 3 do not have the hydroxyl groups coming off of the the 2' carbon. And then deoxyribose, so those are going to be the other three, the ones that lack that hydroxyl group. So A, B, and D. And then the pyrimidines, so those are going to be the ones with single, the nitrogenous bases with singular ring structures. So A and B have double ring structures, so those wouldn't be it. So it'd be C, D, E, and F for the pyrimidines. And I have the 3 pyrimidines that could be present and identified these 4. And that should be it for this week's.